cells. So let's get started on trying to understand what immunity is and what it offers us and how it plays out. So immunis is a Latin term that means exempt, suggesting we are exempt from any sort of disease or pathogens. A pathogen is anything that's going to enter our body and cause some sort of disease or pathology. Generally, we think of these as microorganisms like bacteria, archaea, parasites, protozoa, and certainly viruses. So let's just get a general look at immunity and the history of studying immunity as we move forward in this discussion on the immune system. Between 430 and 404 BC, there was a war between Athens and Sparta, known as the Peloponnesian War. And this war was very tough on the people of Athens because at the very same time, they were afflicted with an infectious disease known as the plague. It's unclear to me if the plague, generally when we speak about the plague, we're talking about a bacteria known as Yersinia pestis. Not sure if that's what was causing this plague. Nonetheless, a lot of people died in Athens due to the plague. And what they realized is that the only people that could provide assistance, health care, or support to these people that were sick were any individuals who actually had this infection and recovered from it. A lot of people were certainly dying from it, but those that recovered could attend to the sick people, the sick warriors, the sick soldiers, and that indicated to the people of Athens that there was some sort of immunity or exemption developed by people who had survived this infection or survived the plague. Moving forward about 1800 years, Dr. Edward Jenner did work with milkmaids. Milkmaids work closely with cows. Cows have a disease that afflicts them occasionally known as cowpox. It is similar to smallpox, much less virulent than smallpox. And what Jenner realized is that these milkmaids could provide health care and assistance to those with smallpox without getting smallpox. Somehow they had developed an exemption or immunity to smallpox merely by being exposed to cowpox when they were working with cows. Now moving forward to the 19th century, in 1878, Louis Pasteur, who was a pharmacist, a chemist, and a microbiologist, was doing work with fowl cholera. Cholera is a bacteria that causes very severe, oftentimes deadly dysentery or diarrhea. So let's take a look at the work that Pasteur did with fowl cholera. Fowl is referring to birds. Pasteur used chickens, and he infected the chickens with Vibrio cholerae, which is cholera. Cholera is the common name for this bacteria. And he grew cholera in a petri dish. He took the cholera and he injected it into some chickens. And all of the chickens he injected it into died. So that doesn't really tell Louis Pasteur anything, but at that point, Louis Pasteur went on vacation and he left this cholera on his lab bench. Could have left it in an incubator or a fridge, but I suspect he just left it out not knowing what to do with it. Now he comes back from vacation and he comes back to that initial growth of cholera that he had. During the time that he was gone, unbeknownst to him, the cholera changed. The cholera had weakened and become attenuated. Attenuated merely means weakened. So then he injected this cholera, the weakened or attenuated cholera, into a different group of chickens, and all of these chickens survived. They did not die. They may have gotten sick, but they did not die. As Louis Pasteur continued doing his work, he grew some more cholera. So I have the cholera here. This is a fresh batch of cholera. I have it drawn in green, whereas back here I had this drawn in blue. And the only reason I'm doing that is to show you that this is a fresh batch. So he grabbed some more chickens. He injected these chickens with this fresh batch of cholera, but he was running short on chickens. So he grabbed some of these chickens, which I have drawn in blue, which were blue over here. And to be clear, the chickens aren't really blue, but I'm just trying to delineate or keep separate the ones that had gotten the attenuated version of the bacteria versus those who had not received anything yet. So these 3 chickens had never been exposed to cholera, whether it was the virulent is deadly or strong. They had never been exposed to any form of cholera, whereas these chickens had previously been exposed to the attenuated version of the cholera. So he injected this cholera into these chickens, and what do you think happened? You are correct. These chickens, who had never been exposed before, all died. But these chickens up here that had previously been given the attenuated version had developed immunity or an exemption to cholera. They were no longer getting sick. They may have been getting slightly sick, kind of like we all do with vaccines, or some of us do with vaccines. But most importantly, they were not dying because they developed immunity to this pathogen, to cholera. And as a result, this is what Louis Pasteur called a vaccine. A vaccine, or at least the early vaccines, were just attenuated or weakened version of some sort of pathogen. He called it a vaccine in honor of Edward Jenner's work with the milkmaids, because vaca refers to cow or cattle. Vaccine, vaca. And that's the early work with understanding immunity and actually understanding vaccines. So Pasteur continued his work, and in 1881, he attenuated anthrax, Bacillus anthracus. He attenuated or weakened this bacteria by heat, by heating it up, and then he injected it into a bunch of sheep. Afterwards, with the same sheep that he had vaccinated with the attenuated version of anthrax, he gave the virulent or very strong version of anthrax that was not treated with heat, and those sheep did not die because they had developed immunity to anthrax. In 1885, there was a young boy who was bitten by a rabid dog. Rabies has a roughly 2-week incubation period where symptoms are not showing up. And rabies is a very deadly virus that gains access to the brain via neurons from the site of the infection or the original bite. What Pasteur did is he injected this boy within the incubation period with an attenuated version of rabies, and it cured the boy. And this is one of the rare instances where a vaccine can actually be given to an individual after they've been exposed to the virus. So these are just examples of the early work with immunity, with vaccines. What we've been describing here, either with smallpox, with anthrax, with rabies, is specific to certain pathogens. Whereas we have our innate or non-specific immunity that has less of a focus on specific pathogens, but a general ability to ward off any harm to our body. So what does innate or non-specific immunity look like? This could run the gamut from our skin, specifically our dead skin cells that lay on top of our epidermis that provides protection, somewhat like body armor. pH deters pathogens, sneezing, tears, coughing, and there's a number of white blood cells that are parts of our innate immunity. But let's look at one component of innate immunity, and that is mucus. So what we see right here are 2 lungs, 2 bronchi, or 1 bronchi going into each lung, and prior to the split into the bronchi, we have the trachea, which is our windpipe. That's allowing air to go into our lungs from the atmosphere. To be clear, pathogens are going to try to enter our body any means possible. And when I say that, I'm not suggesting that any particular pathogen can gain access in any way, but the respiratory system is an avenue that many pathogens try to enter our body. So the mucus here, so right here is the trachea. So this is representing this right here. This is the space that air would flow through down the windpipe or trachea. And these are epithelial cells, specifically pseudostratified epithelial tissue with cilia. The red structures coming off it are cilia. But what I want to point out is in green here is the mucus. And the mucus has a very special, important role, and that's to trap any pathogens or debris that are trying to enter our airway to gain access to our body. The open space of the windpipe, or the open space of any tube, is known as the lumen. So this is the lumen of the trachea. And once again, we see these cilia that are projecting inward towards the lumen through the mucus. So the mucus is somewhat thick, not super thick. It needs to be fluid enough that it can move. And how does it move? The cilia actually propel that mucus superiorly. So once again, the mucus is trapping pathogens, dust, and debris, and the goal is to get that mucus, that bacteria or pathogen-laden mucus, up and out of the airway into the oral cavity, where then there's digestive enzymes that can take care of that mucus, or it can be eliminated. So what we want to talk about here related to immunity is part of the production of this mucus. Like I said, the cilia need to be able to propel this mucus superiorly up and out of the airway. So it can't be too thick that the cilia can't propel it. So let's talk about how this nice fluid mucus is created, and specifically the fluidity part of it. The mucus is secreted by goblet cells and the epithelial tissue. I'm not focusing on that, but I want to focus on the water that's added to this mucus to allow it to move freely up and out of the airway. So right here, we have an epithelial cell. To be clear, this is the trachea, that's the lumen of the trachea, and each one of these squares is an epithelial cell. So I've just blown this up. We're looking at one of these epithelial cells. This right here is the lumen side. So lumen here, lumen here. So we're really looking just at one of these cells on the left side of this image. This is the extracellular fluid side, this is the lumen side, and this is one epithelial cell. So we have a number of channels and pumps here. We have a sodium-potassium pump up here. In red, we have a NKCC, which stands for the sodium-potassium chloride co-transporter, because it's moving sodium, chloride, and potassium into this cell. Sodium, chloride, potassium co-transporter. The chloride then proceeds to move into the lumen via this chloride channel, which is known as the CFTR. That is the cystic fibrosis transmembrane conductance regulator. I know that's a mouthful, but just understand this is a chloride channel that allows chloride to move from the interior of the cell into the lumen of the trachea. It's really important that this chloride moves into the lumen of the trachea, and we'll get to the reason in a moment. The sodium that comes in, we don't want the sodium that comes in to depolarize this cell. So that sodium is immediately going to be pumped out via the sodium-potassium pump. Certainly potassium is going to come in, but that potassium can leave via any number of potassium leakage channels. Just like this potassium that comes in, that immediately leaves down its chemical gradient from an area of high concentration of potassium in the cell to low outside the cell. We know that because that's the baseline we've established when we start talking about physiology. So once again, sodium comes in and immediately leaves via the sodium-potassium pump. Potassium comes in and immediately leaves via potassium leakage channels. Chloride comes in and immediately leaves into the lumen via this chloride channel, the CFTR. So once we have loaded the lumen up and the mucus that's in the lumen, I don't have the mucus drawn here, but once we have a bunch of chloride in here, it's going to be more than two. Sodium is going to chase the chloride. Sodium is going to move actually between cells via what's known as a paracellular pathway. And sodium's chasing chloride because positive charges like to be around negative charges. So the cations are going to follow the anions. Chloride is an anion, it is a negatively charged ion. So because chloride is in here, sodium is going to follow chloride. Now we have a bunch of sodium and chloride in here. And now water along that same paracellular pathway is going to chase all of these solutes. Water, via the processes of osmosis, water is always moving to an area of higher solute concentration. To be clear, if there's a higher solute concentration, that means there's a lower water concentration, and water just moves to an area of low molecular water concentration, but that also means to an area of higher solutes. And this is a classic case of osmosis. So in review, chloride gets moved into the cell via the sodium-potassium chloride channel. It then moves into the lumen via the cystic fibrosis transmembrane conductance regulator, which is a chloride channel. Sodium is going to follow all of these anions, and the water is going to follow all of these solutes. What is this going to do? This is going to produce a very nice mucus that's fluid enough that can be propelled superiorly up and out of the airway with bacteria, with pathogens, with viruses that are trying to infect our body. This is a very classic and important component of our innate non-specific immunity. To be clear, it is not focusing on any specific pathogen. Anything that gets trapped into the mucus is going to be eliminated from the body. Now, why is it called the cystic fibrosis transmembrane conductance regulator? Because individuals with cystic fibrosis do not have this chloride channel. Because they don't have the channel, chloride is not moved into the lumen of the trachea. And to be clear, we find these channels other places in the body, but with regards to the respiratory system, we don't get chloride within the lumen of the trachea. Since we don't get a buildup of anions here, we don't get cations wanting to come into the lumen. So now we don't have anions, we don't have cations, and we don't have anything drawing or sucking water into the lumen, which means we get a very thick, stagnant mucus here that cannot be propelled superiorly by the cilia, and then that mucus just stagnates there, harbors bacteria, and causes infections. So that is an example one of the use of the mucus, and two, what can happen if something goes wrong with any of the apparatuses within these cells, specifically the CFTR channel. Now, we do see CFTR channels within our intestines as well, and an interesting case study with that is that cholera releases toxins within the intestines that actually amplifies or stimulates the production of these CFTR channels within the intestines. Keep in mind the intestines are one big long tube with a huge lumen in it, and it stimulates the activity of many CFTR channels. That is to say, the cholera toxin does. Because there's more activity, more pumping of of chloride into the lumen of the intestines, there's going to be a lot of sodium moving into the lumen, and because there's a lot of sodium and a lot of chloride, there's a lot of buildup of solutes within the lumen of the intestines, so a ton of water chases it, and that's what causes dysentery or diarrhea from cholera infections. So that's just a little glimpse into our immunity, both innate and acquired, and we'll talk more about this in the next video.